So now that we've sort of talked about the structure of DNA, we're going to focus on the replication of DNA. The term DNA replication simply refers to the process of copying the DNA molecule. Now we're going to do this through a process known as semi-conservative replication. The reason that we call it semi-conservative replication is because each strand of the original double helix, the parental molecule, serves as a template for a new strand in the daughter molecule. And we're going to walk through what that process looks like. So here's what semi-conservative replication looks like. The first thing that's going to happen is that the DNA molecule is going to unwind. And when it does so, it's going to expose all of these nucleotide bases. Now you can kind of think of these nucleotide bases as like puzzle pieces that are only going to fit together in a certain way, but they're also kind of puzzle pieces that are sort of attracted to each other. So if we just unzip and open up this DNA molecule, the corresponding nucleotide bases, the little pieces, are going to just fill in as we go. They're going to just fill into the empty spaces in the strand. And what we're going to see is that we now have an exact copy of DNA. If the original sequence was CACTGGTACAGGT, we now have two strands that have that exact same sequence. CACTGGTACAGGT, CACTGGTACAGGT. So this is going to be the end product of our replication. This is why we call it semi-conservative. We just simply open up the original molecule and then we fill in the pieces on both sides, creating a mirror image of the opposite side, which is going to create two complete strands that perfectly mirror the original. So we're going to go through each one of these steps of this DNA replication. DNA replication occurs in three main steps. The first being unwinding, or separation. We're going to separate those two strands of the parental DNA molecule with the aid of an enzyme known as DNA helicase. So DNA helicase is going to be the enzyme that is responsible for unwinding the parental strand of DNA. These separated strands are now going to become the template to create two DNA molecules. Now the DNA is stable as a double-stranded helix, but it's unstable in its single-stranded form. So it's going to require single-stranded binding proteins to bind to those single strands of DNA, help stabilize them. It keeps them from twisting back up. So you can kind of think of this as like a like a double-stranded yarn. If you were to untwist it, you're going to then need to stabilize those separated strands, or otherwise they're going to wind back up around each other. So we use these single-strand binding proteins to help stabilize those single strands. Now once we have stabilized those single strands, we can then start to fill in the missing pieces. We're going to do this with a primer. A primer is going to be DNA replication requires a primer. It's just a short strand of RNA to signal the start of DNA replication. This primer is put in place by DNA primase. Once bound, the DNA polymerase separate enzyme is going to recognize the primer, and it begins DNA replication. Now this DNA replication allows new nucleotides to form complementary base pairs with the old strand, and it's going to connect the new base pairs in a long chain. It also is going to proofread that new strand. So once DNA replication begins, it proceeds in the 5' to 3' direction. Now the parent strands, like we said before, they're anti-parallel, meaning that when they're lined up, one runs from the 5' to the 3' end, and the other is going to run 3 to 5' end. Now, the new daughter strands are also going to be anti-parallel, so one strand is going to be synthesized from the 5' to 3'. This is what's known as the leading strand. DNA can only be read in one direction, therefore it can only be synthesized in one direction. Now, here's the thing. We need to synthesize both sides of that DNA at the same time, and since we can only read in one direction, one strand is going to be read correctly in leading strands, synthesized from the 5' to the 3' end. But the lagging strand must still be synthesized in the 5' to 3' direction, but it must do so in small short bursts as the strand itself is running backwards from 3' to 4'. These small fragments are known as Okazaki fragments. So I just want to kind of take a moment and have you think about this. It's kind of like if you were to read a sentence forwards. So if you were to read this sentence forwards, you would say, parent strands are anti-parallel. Now, if you were to read this sentence backwards, it would be very, very difficult and it would be impossible to read this backwards, starting over here at this far L. You would get a word like Lelarapitan, itna. Lelarapitna. That's not a word. It doesn't make any sense. If you were to copy this sentence down, that word, it's not a word. It makes no sense. So if you were to copy this sentence backwards, if you had to start on the right and copy this down to the left, you would start by jumping ahead to the beginning of the word. So you would jump ahead and go anti-parallel. Then you would jump ahead to the beginning of this word, are. Then you would jump ahead to the beginning of this word, strands. And then you would jump ahead to the beginning of this word, parent. So your sentence would still read parent strands are anti-parallel, even though you had to read it backwards and forwards. So what you would do is jump ahead and then go forwards, and then jump ahead and then go forwards, and then jump ahead and then go forwards. So that is what our lagging strand does by using Okazaki fragments. And this is a fantastic opportunity for you to go ahead and send me some extra credit videos on lagging and leading strands, especially Okazaki fragments. It's going to help you understand just how we're able to synthesize these strands of DNA backwards. So here's again what that looks like more visually. One strand, the leading strand, the DNA polymerase is going to synthesize this strand from the 5' to the 3' end. So it's basically going to read this sentence forward. This DNA polymerase, on the other hand, has it a little bit tougher. He can only read in one direction. So what he's going to do is he's going to jump ahead and then synthesize the strand, and then jump ahead and then synthesize the strand, then jump ahead and synthesize a strand. These short little fragments here, are what are called Okazaki fragments, and they're going to be glued together into one seamless strand by the aid of the molecule DNA ligase. So as we said before, DNA polymerase is going to join the new nucleotide bases to the old strand to make double-stranded DNA. DNA ligase is going to be responsible for gluing those Okazaki fragments together, so we have one long, smooth, continuous strand of DNA. The end result is two double-stranded DNA molecules that are absolutely identical to the parent DNA. So eukaryotic DNA replication is going to occur slightly differently than prokaryotic DNA replication. So we're going to just break down some of the similarities and differences. With eukaryotic DNA replication, we start by forming a DNA replication. The DNA replication can actually occur at numerous points. So here we'll start off by forming what is known as a replication fork. The DNA is going to unwind from its helical form and unzip into two strands. As it is unzipping, DNA fragments or nucleotide base pair fragments are going to start to fill in the missing pieces. So that way, by the time these replication forks or these replication bubbles join together, we have a completed parental strand of DNA. One thing we want to mention is the accuracy of DNA replication. DNA replication is a highly accurate process. The DNA polymerase makes a mistake only about once per 100,000 base pairs. But not only does it only make a mistake once every 100,000 base pairs, it actually has the ability to proofread as it goes, and it can correct those mistakes. So the resulting accuracy for DNA polymerase is only about one error in 1,000,000 base pairs. It is highly, highly, highly accurate. However, that still is not 100% accuracy. It's darn close, but it's not 100%. This is how mistakes and mutations can occur. And we're going to talk a little bit about genetic mutations in our next chapter, chapter 13.